Hello, everybody. It's always nice to be up on a podium and see everybody. I can see if you go to sleep. But first, I want to thank particularly Maribel and Gabriela. I met Maribel 2 years ago, and I told her I really want to see Cuatro Ciénegas, and she made it happen. So that is really amazing, and I'm looking forward to lots of conversations. So thank you, everybody, for listening for the next 45 minutes. So. What I want to do today is, as Gabriela said, go over the arc of what I've been interested in for 20 years. Sometimes there's a logic, sometimes there's not a logic, but that's what I'm going to tell you about, how we understand diversity and how we understand differences in phototrophic communities. So the first question, I'm going to be asking a lot of questions because that's how my mind works. Why should we focus on phototrophic communities? And that is said very beautifully by two folks. One is, and I think this, again, not a word I want to use, and I want to ask how many of you know the word déjà vu? So most of you, so I don't have to explain. You will get a sense of déjà vu between Valeria's talk and mine, and hopefully that will bring up a lot of questions in your mind. What are the similarities and what are the dissimilarities? So going back to this idea of phototrophic communities, what we're really looking at is that 80% of the natural history of life on Earth is exclusively the history of microbial communities. That's a pretty impressive number because it really says we need to focus on these communities. And the other thing is the importance of biofilms. And both of these will figure heavily in my talk today. So I want to remind you, because I'm sure many of you know, they're lovingly called cyanodes, but cyanobacteria as a group is really fascinating because, oops, is there something you can fix here?
It's ready here, it's happening here.
It's okay.
All right. So cyanobacteria, as most of you know, but just to remind you, they've been called life's microbial heroes. I put in the IN because I think they're heroines. They're an ancient phylum that have existed for over 2 billion years. That credits some respect. And not just that, they have also other progenitors of oxygen and photosynthesis. So part of the reason we are here today is the fact that cyanobacteria figured out how to split water and produce oxygen in the atmosphere. We've heard this already, that they actually, because of their photosynthesis, they're found almost everywhere and they're primary producers, both in terrestrial, as we had just heard, and in marine communities. What is really fascinating to me and what got me interested is that in these biofilms in extreme environments, cyanobacteria are incredibly resistant to environmental stress. That tells me two things. One is they figured out how to deal with stress, and second, we may be able to use this feature as the world heats, dries, and is several other onslaughts on the environment. I will talk quite a bit about this, but what is fascinating about cyanobacteria is not only do they do photosynthesis, they can also fix nitrogen, which is another amazing feature because you now have the ability to both do carbon fixation and nitrogen fixation in one organism. Finally, because of all of that, they are really a potential source for doing a lot of synthetic biology. I don't think we've made as many strides as we could, but it's coming. So I'm going to do, sorting out what Gabriela said, I'm going to talk about my life in 2 slides. So if you fall asleep after this, it's okay. I started very small, really just trying to understand a very simple but beautiful phenomenon. And that is if you just streak out Synecocystis, it's a model organism, on an agarose plate, low concentration. You have light coming in one direction. You see these beautiful fingers of cells moving towards the light. And also, and this is for the physicists I just met this morning, they actually make these interesting patterns. The fingers don't all move together. But that, I think we understood, and I'll show you in the next slide, we understood some of the molecular features of cyanobacteria, and I will explain in the next slide why I moved into what I call messy life. because now you go from understanding one organism in all its intricacy to understanding a whole community. And I think the important thing that drives me is what can we really discover? What are the rules? What are the exceptions? And the counterpoint to that is we cannot, although we may like to, go to every environment, do metagenomes, isolate organisms. But the question really is, are there underlying principles? And I would say that's where biologists and modelers and geophysicists have to come together, because otherwise we remain in these silos and go deeper and deeper looking at particular problems without sometimes spreading out. And I want to try and do that. So, you're not really supposed to look at this in any detail, but what it's telling you is many years of work that showed us that in these cells are just about a micron across. They're tiny, and yet they can actually do what I call positive phototaxis and negative phototaxis. They can sense light, and they can move towards it. They can sense too much light or UV, and they can move away. And they have a very sophisticated system that took our lab and many other labs to sort out. There are multiple photoreceptors because they can actually gauge different light qualities. And then there's a complex signal transduction pathway by which all of these photoreceptors talk to these things called type 4 pili, which allow them to move. So this is in one slide again, showing you 20 years of work. So I've done my job. However, the big question that I leave and pose to you is, and that has kept me up at nights, is sure, I understand all the molecular mechanisms of phototaxis, but why do they do this? And that is my feeling was, we cannot continue to work with one guy in the lab feeling very happy. We have to take a leap. And it isn't as easy as that. There were lots of ins and outs, but that's the way I'm going to tell the story today. So, this is the introduction to what I'm going to talk to you about. So this one I think you can pay a little bit of attention to. So this is a place called Octopus Spring. And you can see here something's really important to remember, and it goes back, I think, to some of the points that Valeria was making. The water comes out boiling from this lake. And you can see around the edges, I hope you can, at least if you're at the front, there's a lot of pigmented stuff. Those are the microbes. So that's the one thing to remember, that they cannot grow at these very high temperatures. But as the temperature cools down these channels, you start to get bacteria or archaea. Now, as I said, my sole focus for many years was cyanobacteria. And the fascinating thing here is that the cyanobacteria are only or mainly found in this very stable temperature gradient. You can come back year after year to Octopus Spring, come around here, and these are graduate students and postdocs who've done that, and you can go and get samples here. It will always be at 70 degrees year-round, or you can go to the lower part at 50 degrees. Now, you may be asking yourself, well, why do you stick to those temperatures? And that's because above 70 degrees, cyanobacteria crap out, and then you get archaea. Below 50, all the pesky eukaryotes start to eat the cyanobacteria. So there's this sweet spot, which is just like about a meter, where the cyanobacteria are doing their thing. And that's what I'm going to focus on for the rest of the talk. The other thing that's worth pointing out is, and in contrast to Puerto Ciénegas, Yellowstone is completely protected area. So actually to even get out this, which looks here huge, but it's actually the size of my little fingernail. That's the kind of samples we're allowed to take out. So everything I'm going to talk to you about is from these tiny little samples. That's point number one. Point number two to remember is these biofilms, like many other extreme biofilms, are being blasted by light as it changes through the day and night cycle. That results in changes in many other gradients of many other features such as oxygen and pH. I'll go into that a lot more detail. But what I'm trying to say here is that in this kind of static or apparently static environment, there's a heck of a lot going on. Most folks go to Yellowstone to look at bison and who knows what else, but theirs has bison. they actually, this is an extremely dynamic system, and I'm going to try and emphasize to you how dynamic it is. Obviously, you cannot use your eyes to do this. You need much more sophisticated tools. microscopy and genomics are some of the things I want to talk to you about. So, how do you do this? And very quickly, because this is published and many of you I think are pretty familiar with it. So you start with this core sample that I just told you. You can freeze it away or you can work with it right in the lab. You can grind up everything, which is the so-called metagenome. A caveat here, because we're going to hear I think a lot about metagenomes, is what you grind up is what you get the DNA for. What is not accessible to breaking will never be analyzed. It's a major caveat that most people never talk about. You can do enrichment cultures, and I think I've heard that word here used before. You try and pick out, for example, in this case, cyanobacteria. The important thing on this slide is to remember is we got Synagococcus, these sausage-shaped guys, one that was isolated from the 60-degree part of the mat, and we had another enrichment culture from 50 degrees mat. And this was done by Dave Ward's group at the University of Montana for many years, even before we jumped into this project. So we were kind of here. We didn't have the metagenome. This is the part that excites me the most. I'm going to use this slide to tell you. Over the years, we have tried to get many of these isolates out of the mats. And the reason for that is twofold. One is I think there's a limited number of experiments you can actually do in situ, not to mention the fact that they're many hundred miles away. But also there are certain things, certain parameters and multiple changes that you cannot make easily in the environment. So my hope and dream was to be able to bring these guys, isolates back, and that opens the door to building a genetic system, which in my vocabulary is a very important step because you go from correlation to actually showing what's going on. You can build synthetic communities. If I have time, I'll tell you a little bit about that. You can start to look at the evolution of populations under stress, and you can start to build tools for synthetic biology. And I won't get to all of those today. So here's my first big surprise, and I hope it'll be a surprise to you guys. Here's what happens when we sequence the genome. And all of you know that most cyanobacteria are particularly vanilla-flavored, 3 megabases circular genomes. So here you are looking at OSP on the outside, just false color from about 0 to 3. Inside here is OSA, the high temperature guy. And even if you're right at the back, you will see that there's something radically different between the two. If these were two very closely related genomes, my hypothesis would be that the two false colors would look pretty much the same, right? Because there isn't much difference in what's called synteny, which is the arrangement of genes along a genome. Here you see some craziness going on, right? Which is telling you that these two genomes are not at all syntenic. And my immediate question is this something important? Is it a random happening? And we kind of went down that hole, sort of looking for changes and important things happening here. So, like I said already, the genomes are very closely shared. So you might say that they're the same species. It's arguable. Clearly, they're both phototrophs. They have a lot of similar features. But the synteny is broken almost every 2 kilobases. Operons are often not broken up, and you can understand why. That would kill that particular cell where something happened. There's a very large number of active transposons and repeat elements, and that's also got a lot of hip stories, which I will, there you are, talk more about. And what I'm going to tell you a lot about and haven't discussed very much is evidence of horizontal gene transfer, which I find another very fascinating phenomenon. And about 300 novel genes and pathways, which I'm not going to tell you about. So, to tell you the first story that we again published many years ago, but is very fascinating, was the discovery in these genomes of about a 23 kilobase region, which smelled and looked like a nitrogenase operon. And I was very excited, and I called up Dave Ward and said, I think we have single unicellular nitrogen-fixing bacteria at these high temperatures. And he said, Devaki, I'm sorry to tell you, we've checked and it doesn't happen. There's no better way to anger a scientist than to say something like that. So we went ahead and I've said most of this, but what we tried to do is to do, and this is an important point, trying to do a lot of these experiments in situ, not to bring the isolates back, which by the way we didn't have at the time, but to do experiments in the actual environment. And really the question that is fascinating is, and I'll come to that in the next slide, one of the things that I'm sure many of you in this audience actually already know is that nitrogenase and and oxygen evolution are completely incompatible processes because nitrogenase is killed by oxygen. So as they say, Houston, we have a problem. How are you going to solve this problem? And the way they solve this problem is shown here. It's a little rough and I go very quickly over it, is the idea that at certain times the cell will be doing oxygen and storing both ATP and reductant, which is what the nitrogenase needs, but it's no longer doing photosynthesis, for example, right here. And this was a fascinating thing, only possible because if a postdoc who was willing to take samples between 4 a.m. and 10 a.m., and he found this huge spike in nitrogenase activity. And what I call it is a window of opportunity where what you're seeing is a sweet spot where you have all the requirements for nitrogenase to work, and then it basically dies. And it has to be rebuilt on a diel cycle, which is pretty amazing because it's telling you it costs to have the nitrogen fixation genes in there, but it's worth it for these guys to have it. And the question I want to have percolate in your head is why? And the reason, before it even gets a chance to percolate, I'll tell you, is, and I think, again, going back to, I think, what Valeria might have said, many of these environments are absolutely lacking in almost any nutrients. So if you've got to hack it there, you've got to learn how to hack it. And the cyanobacteria can do that. Like I said, they do photosynthesis and nitrogen fixation. And that's one story I want to tell you. So this is something I've already told you, I don't need to repeat it. But the question that again bothered me is, are we going to make our life understanding of these based on 2 genomes when we know that there's so much dynamic and fluctuation going on? We need to bring that into the picture. And how do we do that? So really the question is, what do we miss when we call Synetocystis 6803 the model organism for unicellular cyanobacteria? And I would argue we are losing a lot, and I'll hopefully convince you of that. So really the question becomes, how do consortia or communities start to deal with stress and perturbation? And I think microbial mats, particularly those in extreme of us, are extremely pretty damn good at doing this, and we need to pay attention to how they're doing this. One thing we know is they do horizontal gene transfer, so huge 23 kb module of nitrogen fixation was acquired from somebody, possibly another cyanobacterium. We know this in the case of phosphonates. We did a lot of work on that. Urea utilization genes are being transferred and exchanged between them. So again, another aspect of the dynamic behavior of these communities, which would be hidden if we only pulled one guy out of this mat and said, he or she represents the entire community. So I go over this very quickly again, simply to make a simple point, and that is that when we started to approach this question of how do we start looking at populations, one thing we did, and this was in the dark ages, we just pulled out regions of the genome and asked how similar and how different are they between OSA and OSB. So just to give you a sense here, we're looking at, this is the OSB here. It also happens to be the most abundant guy. And this is because we took the mat from about 50 degrees. Here is OSA way out there. Okay, so it fits the picture we have of these being closely related, and then there are other guys that are variants on this. But 16S, even though it's widely used, I would argue misses some of these more nuanced images and pictures of what's going on. So if you look at a particular protein org, you now see the picture is much more complicated, and you have all these variants. Again, just to orient you, here is the most abundant guy. It's not OSB. Here is OSB, and way there out in space is OSA. So it's telling you that if you do this kind of genome sequencing just for a particular region, you see that there are all these variants that are not captured by OSA and OSP. I hope that makes sense, but if it doesn't, I'll happily talk much more about it. But the idea really, the principal idea is that these are populations, all of which are slightly different from each other. In my mind, the importance of that is that evolution and stress can actually work on all of them. It doesn't have to pick and choose. And so you might see fluctuations in who's doing well and who's not doing well. And that's something I think we want to do in the future. So essentially, this is to summarize a lot of data, because there's a lot of very great ecological literature that suggests there's a niche, and guys actually then respond to that niche. Or what we are seeing is really what was very nicely called by the science review was mosaic genomes. And I think those two have two very different pictures of what might be happening in these communities. And what we have been doing, and I probably won't get a chance to talk about it, we also have a lot of information now about how these variants spread over time and in the different springs, but I won't have the time to talk about that. So, this brings me to a turning point in my life, that we basically realized we had to take this deep dive into saying we cannot just get away, even though I'm a card-carrying molecular biologist, by just doing what we're doing. We need to get more data. You can use the cowboy approach, or you can use the Oliver Twist approach, but either way, you need more data. And we actually managed to convince folks at the Joint Genome Institute. And if you guys don't know about the Joint Genome Institute, please come and talk to me, because they do really still have major grants to do the kind of stuff that allows you to do very deep sequencing, etc. So what we did in a nutshell is we asked them very ambitiously that we want metagenomic data from two different springs, octopus and mushroom spring, to see if there's any differences. We want to do it over time series, because then you can see what changes are happening. And we also want to see what are the differences across a temperature gradient. So almost as much data as we could possibly hope to get. We also asked for a metatranscriptomic data, and the reason for that is, as I just mentioned, and you're probably thinking about it, is that things will change over a day-night cycle, and we had no clue. We just picked nitrogen fixation as one story. We also did single amplified genomes. I won't talk about that, but there's another reason for doing that. And we did a lot of work on the viromics. This was right between COVID and everything else, so this is a long, extended story. I'm still going to try and stick to 45 minutes. So, my postdoc made this slide, and I find it impossible to go through. But basically, the idea, what she was trying to establish here is the sampling is two different springs. We did, so this is a repetition of what I just said. We did a metagenome over many years. We did both octopus and mushroom, and we did a metatranscriptome, less data there. And we tried to look at it over temperature, like I just said, and over a daylight cycle. And then this line, which now appears as a single line, was really painful. Painful for many, many reasons, and that is that even though we talk about MAGs and we talk about single genomes, it's much more complicated. And as soon as you go away from pipelines that you can run your stuff through, you see that there's lots of gaps in how we're doing things, and they don't address the kinds. I see some nods. It really doesn't address the kinds of nitty-gritty we wanted to get into, and I leave it at that. And we also, and this is a really interesting, I'm happy to talk more about this, how we tried to do this, which was kind of non-standard. And the questions we were asking, which we all ask, is what taxa are present, which taxa are active and when, are community behaviors coordinated, and then something that I've been brought kicking and streaming into is to try and think about heterotrophs, because all my life I've really thought about cyanobacteria. So, to step back a minute, just so that you're following my train of logic, what's really happening is in these mats, and let me just quickly explain, the oxygenic phototrophs, for example, the cyanobacteria, have to stay on the very top. And why is that? They need light. And they need particular wavelengths of light. But below, and so they are releasing oxygen. And so this happens during the day. But in here, there are lots of anoxygenic phototrophs. There are anaerobic heterotrophs. There are aerobic heterotrophs. This is all in there. And what I've done so far is give you the picture from cyanobacteria. But now I want to zoom out, because we are going to get all this data, to say, well, what's happening at the community level? And this, because it can get a lot more complicated, I decided to simplify, is during the day, obviously, because the cyanobacteria are generating oxygen, you have an increase in oxygen, increase in pH, and a reduction in hydrogen levels. And kind of the opposite happens during the night. So what you're seeing then is somehow the day-night cycle is triggering all these behaviors. And when it triggers these behaviors, the question we wanted to ask is who is controlling what? I don't think we know the answer to that, but we have some clues. The more important thing I also want to emphasize, because it'll come back, but I haven't emphasized it in this talk, is these changes between the sun and night happen very quickly. In about half an hour, the mat can go from being superoxide, so 10 times air levels of oxygen. In half an hour, because the heterotrophs are in there, they suck up the oxygen, and then you go anaerobic. So really, it's like a day-night change, and the question is, how are the organisms responding? So, again, this is a horribly complicated slide, but I will give a big prize to anybody who can show me how to simplify this. I will tell you just one thing. Focus really on this part, but this is the answer to how many taxa are there. And I can feel confident now that we know there are about 72 taxa, give or take, depends on how you define them. Some are very abundant. Some are what I call variable, because I can't come up with a better name. And what you're looking at here are different springs, different temperatures. And then you have these rare guys, which we definitely see and we know zilch about. And hopefully 10 years from now we will. But right now we just focus on these very active guys shown by the stars. And almost all of them are either phototrophs or anoxygenic phototrophs. And so what you're looking at here, again, just a very gestalt view, is now what you're looking at. These are springs, orange and red, orange and green. You have the different years, and you have the different temperatures. But this was all done at 60 degrees, because we really didn't have any data. What you're seeing here in the yellow are the most active guys. And here then you have these that are variable, and then these that are rare. Now, so far we made a maybe unfair choice, but we said, well, let's look at the most abundant guys, and let's look at the most active guys, and see if we can make a story out of that makes sense to us. This is something I don't want to dwell on, but to remind any of you who have looked at this, the very critical and obvious way to do it is to look at sentinel genes. For example, here what you're looking at is the eight most abundant guys, and we look at sentinel genes, for example, nitrogen metabolism, motility, phototrophy, fermentation, stadium crop. These are things we actually know about. And one might argue, well, you know about this, so it's a kind of proof of concept that your metatranscriptome actually reflects what you think you know. For example, you can see here in phototrophy, you can see in Silicococcus, a lot of the genes go down at night in this gray region and very quickly go up during the day. So it kind of tells you you're on the right track. But in a way, and so the arguments are, Amanda and I have for a very long time, if you have all this data, why would you look at 10 genes? We could have just done that without doing this complicated metatranscriptome, etc. So what I'll show you now here, again, these are very complex data sets. It's hot, and I can't expect you to follow this, but please follow up on me. So what we're again looking at these 8 major active species. And now what you're looking at in each of these is how they behave over the night period and the day period. And if you squint at this, you'll see certain patterns. For example, let me just show you again, so I can orient myself here. Here's Synechococcus, and you can see in the yellow is the most active. So this is looking at all the genes sort of hierarchically clustered. And you see that they don't have much activity at night, but boom, 6 o'clock in the morning, a lot of genes come on. Okay? So that's telling you about Synechococcus, gave me a sense of relief, and made me trust maybe some of the other guys. And now you see something which we haven't really delved into. Two other pictures I want to share with you. One is Thermochlorobacter, and you can see that it's a huge difference between the night and the day. It almost does nothing at night and a lot during the day. These phototrophs, which are the anoxygenic phototrophs, which I understand a lot less about, actually have much more variable pathways. And that makes sense because these guys can do a multiplicity of both metabolisms and behaviors. And we hopefully can now look into this and try to make sense of that. Synechococcus, I think we know the most about. These, I would argue, although we haven't done that, may tell us more about how they can actually partition their behaviors over a day-night cycle, because we now have this data and we can dig in. Very quickly, one of the interesting things we found is that there are two very abundant heterotrophs which were completely missed in earlier analysis. And what's different about them, without my going into the details, is one seems to have much of its activity during the night, the other seems to have more activity during the day. These are not closely related, but they're from the same phylum. So what's going on? So that's when we could actually dig into this and look more closely, and it seems there's an overexpression of genes that allow this guy, which works mostly in the day, to use things like amino acids and sugars, which are probably being spit out by the cyanobacteria. But these guys can use small sugars, which might be broken down at night. So this is an interesting story to me, that these guys have chosen different ways of behaving based on what kind of sources of carbon they can use, and they've partitioned out. So anyway, I'll skip this one now. So this is a quick summary before I switch to defense, and I don't know how much more time we have.
I don't know either. You have 10, 15 minutes.
Okay. All right, then you can read this. So I told you already that there's these highly active 8 genera. They all respond to day-night cycle, and different sets of genes are highly expressed at different times. But the real question, I think that's most fundamental to this, is who is driving this? Is it the cyanobacteria that are driving it, producing excess sugars or carbon, and everybody else kind of follows? What is the role of the anoxygenic guys? And the reason I say this is, in most biofilms, not all, you find that the cyanobacteria are often with other guys. And the question is why? I would argue that it's certainly metabolism, but it may also have to do with spatial formation of biofilms. And if I get a chance, I'll talk to you a little bit more about that. So, pause here, think about any questions and we'll come back to them. But I want to switch gears entirely to something that we came across in serendipity. We found that the cyanobacteria, which is what we focused on, have these Cas-CRISPR systems. And it suddenly alerted us to the fact that you're not living in happy coexistence just with bacteria. You're being attacked all the time by viruses. And we completely ignored that till we were forced into thinking about it. And so just to give you a very quick summary of what I think the state of the field right now is because of these culture-independent ways of getting at viruses, you can sequence anything you want, anytime you want. That is fantastic. However, there's a major caveat, and that we only have looked at a very small percentage of these viruses in the world, and we know even less about the many layers of defenses they have. But it tells me that virus behavior, what viruses do to the host, is an extremely important part of biofilms because they're stuck close to each other. A cell dies, it releases virus. And somebody has to take up the slack for things like that. And we have used the CRISPR immunity system to try and get to some of the questions of host-virus interactions. And we think that there are actually many abundant and novel phage, and we're trying to get to that. And this is something I will share with you if you come find me later. There's some evidence, because we have the transcriptomic data, remember, over a day-night cycle, we can try to look at what the viruses are doing. And there are some viruses that seem to have more activity during the day and less at night. What does that mean? I don't know yet, but that's just an observation. So what is it that's so beautiful and elegant about the Cas-CRISPR system? It is that as the Cas-CRISPR system is used, it's basically an adaptive immunity system. It's picking up bits and pieces from the phage pool and bringing it into the host, such that that host then remembers that it's like a calling card. The next time the same phage attacks, kaboom, it's dead. So this is an incredible system, but it's also a very interesting system because as these new spacers come in, it's kind of telling you who are the phages that are attacking these guys. So if you look over a day or over a time series, you can see, are these spacers changing? How fast are they changing? And that to me is a really fascinating question because we can now, because we have these oodles of data, we can look not just at cyanobacteria, we can look at everybody who's there who has Cas-CRISPR systems. And it's something I would love to talk to anybody here more about. So, I'll skip now because time's getting tight. One of the things that this is a lovely little story. It started with the fact that we had all these metatranscriptomes, right? I just told you 2 hours over every 2 hours over a 24-hour cycle, and many other people. So these guys, Uri and I shouldn't say gang, group, went and scraped all metatranscriptome data from whatever was available. And they looked for RNA viruses. And the reason they look for RNA viruses is for most other viruses, there are only a few proteins that are genes that are sentinel. For RNA viruses, there is something called RNA-dependent RNA polymerase. So you can look at the metatranscriptome from anywhere and pull out the RNA-dependent RNA polymerase. Now, why in hell am I telling you all this? It is because when they did that, what they found was, I've got these a little bit backwards, but anyway, what they found was there were RDRP genes in the metatranscriptome from the hot springs. And that was a real surprise. And the reason for that is, I don't know how to go backwards, I'm just going to tell you. The reason is that most RNA viruses that we know about either attack humans, animals, or plants. In fact, SARS-CoV-2 is one of those. They're either double-stranded or single-stranded RNA viruses. Almost no evidence of RNA viruses attacking prokaryotes. So this was interesting, but it was more interesting for something we discovered. I wouldn't say Mary discovered really, is he found, so what they did was, now they took these two proteins of the RNA virus, the so-called RDRP region and the capsid region, and they asked, are there any regions here that match the spacers in any of the metagenomes we have, right? So trying to make a correlation between the RNA virus and their host. And kaboom, they got a match. They got a match between the spacers in rosiflexus, which is this filamentous and oxygenic guy, and its spacer matches to the RNA virus. So in one swoop, we had discovered both the fact that there are RNA phages that potentially infect rosiflexus, and that these spacers link them back to the host. Okay? So that opens up, in my mind, a lot of different questions. The question is, how are these changing? Why is it that rosiflexus has an RNA virus, and to our knowledge others don't, but we don't really know that that's true. But it suddenly opened a window for us because we now can grow rosiflexus in the lab. It hasn't been easy to infect them, but we can now start to do, which is my dream project, is to understand how does viral evolution work. And we do it through the lens of these CRISPR spacers. again, I think in the interest of time, I won't bother you with this, but we basically have been working with Simon Rue's group and Clement Copeland to basically now do the first analysis of all the hot spring viruses, what they are, and again, here you're looking at the DNA viruses, there are a lot fewer RNA viruses. We don't even know in most cases who the hosts are. So, just in the last few slides, I just want to say that even what I've told you, I think if you're really clued into all of this, you'll think you're still missing a lot. And so what we're trying to do is this new method, which is, so let me backtrack a second. Short reads, which is what most of our metagenome sequencing was about, really is crappy if you want to do across long lengths. And we missed that completely. So we linked up, Amanda got this very nice prize of doing deep sequencing with BiGEvo. So now what you can do is, when you get long reads, these CRISPR spacers, for example, you can match them to the host. And then something that she's trying, which is kind of almost science fiction to me, is you can start to link the DNA of a host with its phage through this method called proxy meta, which is basically ligation of two pieces of DNA that are very close to each other, and then you sequence out and you try and see correlation. It's, I think, an art that has a long way to go, but it's just to tell you that we're trying to think of as many ways that we can sort of tease apart, I believe, a very complex system. All right, so this is what we have. I was told I should not show this slide because the way the analysis is done is not good. However, we are redoing the analysis in a lot more detail, but enough to say that with this proxy meta data, you can begin to see, for example, rosiflexus. I hope a name you won't forget. This is the one which is a filamentous guy. It has hundreds of spacers. We actually don't know why, but it has allowed us potentially. So here you're looking, this is the host, and you can see here by this code, this is linking it to various viruses. So it's telling us the proxy meta data might work. The question is, how real is it? Because this kind of method is riddled with artifacts. But as we do more and more about this, I think we'll get better at this. And it's a new way of thinking about host-phage interactions. Okay, now, wait, I must show this slide. So we go back. So I hope you can see this. So one last little piece of vignette for anybody interested in phototaxis. You can see chloroflexus, which is related to rosiflexus. Here is light coming in from the bottom. You can see, or almost see, rosiflexus just spreads in all directions. It's motile, but not phototactic. Here's Cinnacococcus, which I already told you is phototactic. And you can see here again, here's the timing of the whole thing. They're moving towards light. What's fascinating is now, if you make a synthetic community of chloroflexus and Cinnacococcus, you see these guys really tracking along these filaments. And to me, that's so fascinating because it says that if you're trying to make a biofilm under flowing water, you better grab onto somebody fast. And it might be an advantage both to chloroflexus and to Cinnacococcus. And well, let's get this. So really, again, that's just a snapshot. We just published this, so it's available to anybody who wants to look. Is the idea, as I said, to use synthetic communities. But I want to end with a kind of a rider to all this. We've talked about cyanobacteria as a great thing, doing a lot of interesting things. They have a very dark side, and that's here. Cyanobacteria, because they can do all these things, if they're limited by either nitrogen or phosphorus, you put it in with fertilizer, golf courses, whatever you have, and you get these massive algal blooms. I'll say personally, California almost never had this, and now with climate change, etc., these events are getting higher and higher everywhere. We've got to learn to deal with them. And microcystis is one of the guys that's really causing a lot of problems. And it's another angle to this whole story. They also do not live alone. They also have a ton of viruses. So it's another story from another angle, but telling us how to think about communities. So this is really all I told you about. I think I told you enough about this, but just to say that can we start to begin to model community behavior in this very particular system? How common are mosaic genomes? Are they really common or are they really uncommon? Are there really spurts of transposon activity and then it dies away? What's the role of phage and viruses? And to me, one of the most interesting questions is, how do these community systems evolve? Are they static for a while? Are there a base of guys who can do their thing and then other ones just hang on? Or is there a succession even in this? And we can start maybe to look at that in synthetic communities. We're also starting to work a little bit on summer tolerance. I won't tell you about that. These are the cast of characters. None of this would be possible without them. Particularly Amanda, who has done a lot of the work I showed you. Freddie did the phototaxis work. These guys have gone back to Spain, but they helped all of them. Tesha's working on the algal blooms, and a bunch of other guys, and many, many collaborators. So, future directions, etc., we can talk about. I just want to say here that I'd be happy to discuss it. I'm around all the time. I neglected almost all detail, which is very annoying in a scientific talk, but I decided to make that choice. But I'm happy to show you all the data. And I want to end particularly with a tribute to Valeria. Literally yesterday when I was getting on the plane, I happened to look at my emails, and there was something from Science, and it was an interview with Kent Redford. And you were at that meeting, I didn't know that. And they said, what action do you want to see come out of the World Conservation Congress? And this is his answer, which I couldn't believe could be more tailored to the conversation here. He said, it would be wonderful to see this group, I don't think he was talking about this group here, but we could do that. To visit places which are valued for their microbes, Quattros enegas, hot springs of Yellowstone, and hydrothermal vents. And basically to make the point that we need to make the world aware of the losses that we don't even know about. And that's something I've never really given serious thought to conservation. But listening to this and seeing what's happening to the world, I don't think we can shy away from doing that. So that's where I would like, I think, to end. Almost on time, if there's. And thank you very much for listening.
Hi, I loved your talk. I wonder if all that recombination you find is because they are on the edge of their niche. They are on the border and they need more diversity in order to cope with that.
Yeah, and I think that's a fundamentally interesting question, and we can try and test that by saying, do other guys who are rare or not doing much not doing this? And the other idea that I've heard before is that there are bursts of transposon activity over time, and then they kind of dwindle away. We don't know that. But the time series might allow us to answer that. But I think it's a fundamental question.
Well, thank you for this amazing talk. I have maybe a crazy or nonsense question.
No crazy questions.
But when I saw your metatranscriptomic result, I was thinking about something like epigenetics process, and maybe there is something like help to the bacteria to up and down these genes from day and night.
Yeah, and that's an excellent question, because again, like I said, I think what people do is they look under the searchlight, right? We can do metagenome, we can do whatever, and that's where we look. We've never looked at very few studies on epigenetics in bacteria, but they do happen. And what I wanted to do with the Proximeta and HiFi, we could have done that, but there was some snafu and we couldn't, which would have been our first indication of whether these changes in epigenetic, expression profiles might be linked to something like that. But I think that's the way to go. Not trivial, but the way to go.
Thank you.
Oh, you have another question? Okay, yeah, let's do that. Last question.
So thank you for your nice talk. I wonder if you have hypothesized what do you have in mind you will find related to the RNA viruses abundance or particular species? What do you have in mind? And the other question is that if you believe that it is correct to try to infer those virus host interactions without performing experimental validations such as CRISPR-Cas.
Two perfect questions. The answer to the RNA viruses is we found them by serendipity. We weren't even looking for them, right? They were just in the metatranscriptome. So if you want to be serious about this, I think, and we've been trying, except we haven't really had a plan to go back to Yellowstone, is to really enrich for RNA viruses, and there are ways to try and do that. So that's the answer to your question. So there were maybe many more, and there was an early work by a group in Montana suggesting that there were RNA viruses. There's a report, I think, of these giant viruses. So I suspect there are other variations. This is just what we have. The question about the experimental is actually something we've struggled with a lot. Because even Neri and his group, who showed this very nice correlation of spacer matches of rosiflexus to the RNA viruses, I think the conventional wisdom in the viral world is that's not enough. We do need to build a system. But building a system is a tremendous amount of work. I mean, getting rosiflexus to grow has been non-trivial. We have to get the viruses out of Yellowstone and functional to be actually able to show that. And if you actually think about it, if you read the viral literature, there's a handful of host phage stuff that work. The good old T4, T7 in E. coli. You go beyond that, it's not matter.